Do you know about the Monoacyl Glycerol Pathway? Do you know about the Bergstrom theory? It's in the topic of digestion and absorption of lipids. Let's discuss this. First, we will discuss digestion of lipids. Dietary lipids, the fats and oils we consume, can be categorized into two main sources, animal fat and vegetable fat. Triglycerides are the dominant form of dietary lipids, making up to 90% of what we ingest. The remaining 10% is combination of cholesterol, cholesterol esters and phospholipids. The average Indian diet typically includes 20 to 30 grams of lipids per day. Digestion initiates in the mouth, but it is minimal there due to brief food retention time. Lingual lipase produced by sublingual glands breaks down the short-chain triglycerides at their first and third carbon atoms, yielding diacyl glycerol and monoacyl glycerol with fatty acids. These short-chain triglycerides are found in milk, butter, ghee, and coconut oil. Lingual lipase is particularly crucial for newborn infants. Next is gastric digestion of lipids. Gastric lipase, produced by the chief cell within the fundus of the stomach, initiates the hydrolysis of triglycerides. Secretion is stimulated by gastrin. Its digestive capacity is limited due to the acidic environment of stomach. As a result, only approximately 30% of lipid digestion occurs. The majority of lipid breakdown and absorption takes place in the small intestine. Next is digestion in small intestine. The small intestine is where most fat digestion happens. Two important juices help. Pancreatic juice. This juice has enzyme that breaks down fats, and bile made by liver. Bile salt present in the bile breaks down the big fat droplets into smaller ones by reducing the surface tension. It is called emulsification. This makes it easier for enzyme to work. Hormones like gastrin, cholecystokinin, secretin, and vasoactive intestinal peptides stimulate the secretion of these juices. Somatostatin inhibits the secretion. Lipid digestion and the role of enzymes. Lipid digestion primarily involves four enzymes secreted by pancreas: pancreatic lipase, colipase, cholesterol esterase, and phospholipase A2. First, we will see action of colipase and lipase. Colipase is produced by the pancreas in an inactive form zymogen and activated by trypsin. Its crucial role is to bind to lipid molecules at the interface between oil and water, creating a suitable environment for lipase activity. Pancreatic lipase efficiently breaks down triacyl glycerol into smaller components. It initially removes a fatty acid from first carbon of glycerol, resulting in 2,3-diacyl glycerol. Subsequently, it cleaves the fatty acid attached to the third carbon, yielding 2-monoacyl glycerol. Since lipase cannot act on second carbon, another enzyme called isomerase repositions the fatty acid to the first carbon, allowing lipase to complete the breakdown into glycerol and 3 fatty acids. The final products of triacyl glycerol digestion includes primarily 2-monoacyl glycerol 72%, 1-monoacyl glycerol 6%, and a mixture of glycerol and fatty acids 22%. Cholesterol esterase cleaves cholesterol esters to produce cholesterol and free fatty acids. Phospholipase A2 acts on phospholipids to form lysophospholipids and free fatty acids. I have made a full video on action of enzyme phospholipases. You can click here on the card and watch it. Now we will see absorption of lipids. Short and medium chain fatty acids are present in milk, butter, ghee, and coconut oil. Coconut oil mainly having medium chain fatty acids. They are directly absorbed from the intestine to the portal vein, and from portal vein it is taken to the liver and are immediately utilized for energy. Now we'll see Bergstrom theory of lipid transport. Beyond its role in emulsification of fats, bile salts play a critical function in lipid transportation. According to the Bergstrom theory, Bile salts combine with digestion products like monoacyl glycerol, cholesterol, and lysophospholipids to form structures called mixed micelles. These micelles are significantly smaller than the emulsion droplets. They have a spherical shape with a hydrophilic outer layer and hydrophobic core, exhibiting amphipathic properties. Micelle formation is essential not only for lipid transportation but also for the absorption of fat-soluble vitamins A, D, E, and K. These micelles are absorbed through the microvilli of jejunal mucosa. Following absorption, bile salts are returned to the liver for re-excretion known as enterohepatic circulation. Now we'll see monoacyl glycerol pathway. Within the intestinal mucosal cell, long-chain fatty acids undergo activation by an enzyme called thiokinase or acyl-CoA synthetase. These long-chain fatty acids are converted to acyl-CoA. This process requires energy in the form of two high-energy phosphate bonds. Two molecules of activated fatty acids then combine with 2 monoacyl glycerol to form triacyl glycerol. Cholesterol is also converted to cholesterol esters, and lysophospholipids are transformed into phospholipids again. Importantly, these newly synthesized lipids differ from the dietary lipids. Free glycerol absorbed from the intestine cannot be directly reused to create new lipids, as it enters the bloodstream. However, intestinal cells can produce glycerol phosphate from glucose and subsequently attach 3 fatty acids to form triacyl glycerol. Now we'll see Kylo Micron formation and transport. Kylo Microns are the transport vehicle for dietary lipids, exogenous lipids. They are composed of a hydrophobic core containing triacyl glycerols and cholesterol esters, surrounded by a hydrophilic outer layer of phospholipids, cholesterol, and apoproteins ApoA1 and B48. This structure enables lipids to be transported through the water-based environment. Due to the large size, chylomicrons cannot directly enter into blood capillaries. Instead, they are transported via lymphatic systems, eventually merging into large veins in the chest, where they enter into bloodstream. Chylomicrons circulate through the bloodstream and are acted upon by lipoprotein lipase present at the endothelium of blood vessels. This enzyme breaks down the triacyl glycerol within the chylomicron into fatty acids and glycerol, allowing these smaller components to be absorbed by peripheral tissues. As triglycerides are released, the chylomicron transform into a chylomicron remnant after acquiring additional apoproteins ApoE and ApoC from HDL. Fatty acids are taken up by tissues like liver, skeletal muscle for energy production by beta oxidation, and by adipose tissue for storage. Now we will see abnormalities in digestion and absorption of lipids. There are 4 types of abnormalities. Number 1, steatorrhea. Number 2, chyluria. Number 3, chylothorax. And number 4, Collithiasis, means gallstone. Steatorrhea or fatty stool occurs when daily excretion of fat in stool is more than 6 grams per day. Steatorrhea can arise from various causes. Exocrine pancreatic insufficiency due to conditions like chronic pancreatitis, cystic fibrosis, or pancreatic tumors can impair lipid digestion. Bile acid deficiency caused by bile duct obstruction by gallstones, pancreatic tumors, and large lymph nodes, or liver diseases like primary biliary cholangitis. These affects lipid absorption. And disease of small intestine such as celiac disease or tropical sprue can interfere with lipid absorption. The type of fat excreted in stool depends on the underlying issue. If the problem lies in lipid digestion, like pancreatic insufficiency, undigested fats appear in the stool. In contrast, if the issue is lipid absorption, like bile duct obstruction, celiac disease, digested fats are excreted. Next is chyluria. Chyluria is a rare condition where lymphatic fluid rich in lipids leaks into the kidneys, causing the urine to appear milky white, particularly after meals. This occurs due to an abnormal connection between the lymphatic systems and urinary tract. A common cause of chyluria is Filariasis, a parasitic infection caused by a worm Wuchereria bancrofti. These parasites obstruct lymphatic vessels, leading to increased pressure and diversion of lymphatic fluids towards the kidneys. Next is chylothorax. Chylothorax refers to the accumulation of lymphatic fluids in pleural cavity. This occurs due to disruption in thoracic duct or its branches, allowing lymphatic fluids to leak into the pleural space, causing pleural effusion. Last is Collithiasis. Collithiasis refers to formation of gallstones within the gallbladder. These stones are composed of various substances including cholesterol, bilirubin and calcium salts. There are three main types of gallstones: cholesterol stones, pigment stones and mixed stones. Gallstones can cause significant discomfort when they obstruct the bile duct, leading to biliary colic, a severe pain in the upper abdomen. Several factors increase the risk of developing gallstones, often summarized by the 5 F's: fair, fertile, fatty female in 40. Chronic collithiasis can also increase the risk of gallbladder cancer. So this is all about the digestion and absorption of lipids. Hope you get the value out of it. If you do, then please like and share this video and also subscribe my channel. Thank you.